In the case of seed samples, you're going to need to grind them into a fine flour in order to submit a sample to the lab. You could also do this for leaves, but leaves have other options such as biopsying or cutting, which is less labor intensive and less messy and has some advantages. So we suggest that for leaves, but for seeds, you're going to need to grind them up. And so you can have two sorts of scenarios. One where a sample's been collected from the field and it has a defined number of seeds in it. In this case, you're going to grind all of them. You can also have a situation where perhaps for a genetic reference sample, a much larger number of seeds have been submitted, and in this case you'll want to count out the number of seeds that's indicated in the protocol, which for a inbreeding pure line crop like bean is 20, and then return the rest of the seeds to the sample. For the grinding procedure then, the seeds that are going to be ground will go into your cup. This is sort of a food blender. This happens to be a brand called Ninja, but there's lots of other brands out there. Typically use a small cup for these small types of samples. It works better for grinding, and then there's also less to clean. So here we would be using all of our sample because this is coming from the field. You know, your crop could be another crop. This is just an example with bean. So closing your assembly, make sure that you don't cross thread it. And then for grinding, you'll want to pulse it. So grind for 5 seconds, let it rest for a bit. So, and you can see that material then comes down, and you do it again. And you're going to want to pulse it about 10 times. Now, sometimes material gets stuck up here on the edges, and you can then tamp that down a bit so that make sure that it gets ground up. And what you want it to be is eventually a pretty fine flour. So you can see there, you're getting pretty fine flour. We'll just pulse it a few more times. So now that that's a fine flour, tamp it down. And you'll want to transfer that back into your sample packet. So if you have good quality sample packets that are paper, you're not going to get leakage out of the sample packet, so you can pour that back in. If you have a concern about your sample packet and that you're going to get leakage of the flour out of it, and that could result in contamination of other packets, you may consider transferring it into a plastic vial instead. So in this case, of course, you would need to transfer the information here for the identifier onto the plastic vial. This is extra time, it's extra materials. So we recommend using good quality packets where possible to ensure that you can just put that sample right back in. Transferring sample also into a vial, this can be a little bit messier. You may wish to use a funnel, but then that's one more thing to wash. And again, transferring that homogenized ground sample as much as possible into your container that's then going to be the storage for the curated sample. Now, for the lab, you're going to take a scoop out of this material and put it into a tube, and we'll show that in another video. This, of course, is dirty, and so now needs to be washed so that you don't get contamination in between samples. And here you can see the way that it's gummed up. This is actually because I did not thoroughly dry it after I had washed it. So you want to make sure that in the washing procedure you thoroughly dry your cup and your blade assembly before moving on to the next sample.